My name is Lucas.
I'm Kaylee.
I'm Carter.
And this is the Fall 2022 Queen Conch Directed Research Project on Microplastics.
The queen conch is a marine snail in the mollusk family and is an important resource for the commercial success of fisheries here in South Caicos. To provide some background info, microplastics are small pieces of plastics less than 5 millimeters in length and can be found throughout the water column. These particles pose potential health hazards to both animals and humans.
These microplastics in the ocean are eaten by animals throughout the food chain, making these plastics abundant in many seafoods. It starts with animals that ingest these microplastics by accident, often mistaking small plastics for plankton or zooplankton.
This topic is important because humans locally consume queen conch here in the Turks and Caicos Islands, and we're pursuing this research project to find out whether microplastics are more prominent in the fecal matter of conch in both the windward and the leeward sides of the South Caicos Island.
The windward side of an island refers to the side the wind comes from, while the leeward side refers to the side which faces away from the wind. We are also determining if there is a difference in microplastic abundance in conch meat and conch feces.
This method of measuring microplastics is a non-lethal way to establish the presence of microplastics in queen conch.
In order to get to our end result of physically measuring our microplastics, we followed a 5-step procedure. We would first collect the conch in 3 different ways. Second, we would hold them in tanks. Third, we would collect the conch fecal matter. Fourth, we would chemically separate the microplastics for observation. And fifth, we would standardize our data and get our final results.
As mentioned before, this study took place on 2 locations for the windward and leeward sides of South Caicos. The 2 windward locations were Family Beach and Long Beach, and the 2 leeward sites were East Harbor and Middleton.
The first of 3 ways our group collected conch was through snorkeling, where we go into the water with these measuring tools called transects and measure 2 distances and count how many conch there were in those given areas, as well as take note of habitat. Later, we would collect 4 conchs in nets to bring back to the center and collect their feces.
The second method of conch collection was through manta towing, where a board was attached to a rope pulled behind a boat. 2 people held on and wrote data on slates consisting of the number of conch found and habitat.
The third method of data collection was scuba diving. Similar to snorkeling, we would collect measurements by laying a transect and taking note of the habitat, how many conch we find, and later collect the conch and bring them up to the boat.
In the boat, you would take 2 measurements, one of the shell length and the other the lip thickness, since the shell lip thickness is an accurate way to measure the maturity of conch.
After we collect the conch, we bring them to the lab where we hold them in tanks for 4 hours. We wait for them to poop and collect the fecal matter.
Once we do this, we put the conch feces in a jar and filter out the seawater. Then we add 30% hydrogen peroxide to dissolve the feces so that we only have plastics remaining. This sits for about 24 to 48 hours. We filter this again and add highly concentrated salt water and let that sit for 24 hours to get the microplastics to float to the top. Then we filter this out one more time to get the remaining microplastics onto the filter.
After getting the filters, we count the number of microplastics present on the screen streaming from the microscope in order to determine the amount of plastic present in each gram of conch feces.
After all of this data collection, we determined that there was no significant difference in the microplastic abundance between conch on the windward side and the leeward side. This suggests that microplastics are found in conch, regardless of the site collected from. Another thing we measured was the size of microplastics across the sites, and we determined that the microplastic sizes were significantly larger on the leeward side of the island as opposed to the windward side.
In this experiment, we found that there was not a significantly higher frequency of microplastics in the fecal matter than in the conch meat. However, what we did find was that the average microplastic length in the meat was smaller, while the fecal matter samples were larger. Of the microplastics we found, we categorized them into 5 different categories: fibers, foams, films, fragments, and microbeads. Our results showed that microplastics were found all throughout the fecal samples, tissue, and even seawater samples. It's sad and scary to say that microplastics are present throughout the ocean and its organisms, and the number of microplastics is increasing. This issue is still pressing since humans in the Turks and Caicos Islands heavily rely on conch for consumption purposes. Research on microplastics to human health is still being assessed, and studies suggest that the impact on humans is widely negative to our health.
Our hopes for the future are that there are more studies focused on the negative impacts of microplastics on human health, as well as more studies looking at the differences between windward and leeward conch that further validate our results. Understanding the potential impact of plastic distribution on windward and leeward sides of the island can aid in management regulations.
This is the Fall 2022 Conch Microplastic Research Project, and thank you so much for watching.